Hi, everyone. Thank you for joining us for today's presentation entitled Highly Multiplexed Digital Spatial Profiling of the Tumor Microenvironment of Head and Neck Squamous Cell Carcinoma Patients Treated with Immune Checkpoint Therapy. It's my real pleasure to introduce you to Arutha Kulasinghe. He's a Peter Doherty NHMRC Early Career Fellow at the Queensland University of Technology here in Australia. For a complete bio of Arutha and all our speakers, please visit the tab at the bottom of your screen. Now, if you have any questions during the presentation, please submit them in the Q&A box there on your screen, and our speaker will respond back to you via email. So please join me in welcoming Arutha. I'll turn the presentation over to him.
Hello, everyone. So my name is Arutha Kulasinghe. I'm an NHMRC Early Career Research Fellow from the Queensland University of Technology, and I'll be talking about some of my spatial proteomics work in head and neck cancer patients that received immune checkpoint therapy. So this work started off, so my background is in looking at circulating tumor cells. So these are transient metastatic cells or metastatic precursor cells that are found in the blood of solid tumor patients. They're typically found as single cells or cell clusters, and I was really interested in these rare cell populations in the blood. And obviously they'll go on to form distant metastases in the lungs, liver, and brain. So we used a number of approaches to capture these circulating tumor cells, as described in the slide including positive selection. So we used FDA-approved EpCAM enrichment technology as well as negative depletion to capture these rare cells. We also looked at filtration-based devices and microfluidic devices to capture these rare cells from a blood sample. And these are some of the technologies we looked at, and we looked at the plasma as well for clinically actionable mutations. So when we were characterizing these circulating tumor cells and phenotyping them, we found that PD-L1 was frequently expressed in these head and neck samples, and that was really interesting to us, and we wanted to look at it in a bit more depth. And we also performed ex vivo culture in a number of these samples. So this is one of the key slides where we found that in circulating tumor cells, PD-L1 was frequently expressed, and it was expressed in head and neck and non-small cell lung cancer. And this is compared to a number of cell lines. Obviously, PD-L1 is dynamically expressed in the tumor, so we wanted to find a more realistic or real-time snapshot of the PD-L1 expression in CTCs, and we thought that this might be a way in which to do that. And this is just some survival metrics at the bottom right-hand corner showing that patients with CTCs had a poorer overall survival in head and neck and non-small cell lung cancer. So that's, I think, the time when Jim Allison's work came out, and the work in CTLA-4 and the science papers that came out showed that in a certain subset of patients, they did really well with immune checkpoint therapy. And this was really driven by melanoma, but head and neck and non-small cell lung cancer, as you can see, were were a subset of patients that benefited from this therapy. So because we were working in this space, I really wanted to go back and look at the tumor of these patients and see what was going on, not just at the PD-L1 expression level or PD-1 levels, but other tumor microenvironment cells as well. So there were a number of biomarkers in this space, and TMB, or tumor mutation burden, was one of them. So on this scale here, you can see that head and neck and non-small cell lung cancer sit in the sort of midpoint of this graph, and high TMB for head and neck is about 10.3 megabases per mutations per megabase or more, and then this is being refined at the moment, and it's sort of being led by melanoma and the data in the first line setting. There, for head and neck, it is given in the metastatic setting, but there is it is now being looked at in recurrent head and neck patients in the first line setting. So this comes on to the tumor microenvironment work that I'll be talking about in this talk in a greater depth, and actually looking at this spatially and getting that level of depth of information, not just expression data at the global scale, but in looking at different regions within the tumor microenvironment was really, really key to moving this work forward. And we were able to do that using the nanostring digital spatial profiler in a pilot study. And we're expanding this work at the moment. So really looking at the microenvironment versus the tumor, the stroma, and comparing it to normal regions to see what was driving these cells. So this is some of the therapies that are given for head and neck, or the progression of it over time. And you could see that in the mid to early 2000s, when PD-L1 was found to be overexpressed in head and neck tissues, and then there was iterations of Pembro and Nivo that came out in the last couple of years, and then where the field is heading now with a number of immunotherapies being developed in this space. So this is the technology we use to profile a number of head and neck tissue samples from patients that received IO therapy. They were recruited to Checkmate studies, and it was a preliminary study. I'm sure the audience is aware of this technology. So there are antibodies. So we looked at the proteins initially, and now we're doing the RNA. So we looked at the protein target. So we were interested in profiling these tumors using the 40-plex immuno-oncology panel from NanoSpring, where the antibodies bind to a UV photocleavable oligo, and when regions of interest are chosen, the signal is liberated, and that can be counted. And there's a number of different data normalization. So I think this is a key slide for a lot of this work. I think it speaks to a lot of this new technology and single cell sequencing, and then the data normalization is becoming increasingly challenging. But I think there are ways in which we can better assess our data. So there are a number of different ways this data was normalized. So ERCC normalization, nucleus normalization, area normalization, and a number of housekeepers include, and signal to noise normalization for each data set. So we were able to measure across the ROIs and within the ROIs if there were tumor or stroma. So these are a number of, just a snapshot of what we do. We get an H&E section. This is assessed by the pathologist to demarcate the tumor and normal region. We tried to do whole exome sequencing on these tumors, but there were limitations with how much DNA was available from them. From one of them, we were able to get enough DNA to do a whole exome, and that patient had a TMB of 5.7 mutations per megabase. So this is your typical H&E stain on the top, and then in the middle, what we can see is a visualization marker stained by the DSP, and stained and imaged by the DSP for pancytokeratin, CD3, CD8, and DAPI, where we would then compare that staining to the H&E and then choose regions of interest. So you can see a number of regions of interest. For these, it was 12 regions of interest per tissue section. And then on the corresponding slide below, those are the regions of interest at a high magnification. So we did this for 8 tumors, 8 HNSCC, so head and neck squamous cell carcinoma tumors. So this is a high resolution image of one of them. So you can see that this has been stained with pancytokeratin, CD3, CD8, and DAPI. And then these 12 regions of interest are selected, and then those are profiled at 40-plex. So the visualization markers are key to choose what you want to profile. So ideally, you'd want to do more if you could. So I think we are looking at doing 24 and expanding this out even more. So this is just some of the data from a head and neck patient that was T4N0, left mandible P16 negative, so HPV negative. This patient had multiple rounds of chemoradiation and surgery and was given immunotherapy, so nivolumab in the metastatic setting. So we had the diagnostic biopsy tissue, we profiled it using the DSP, looked at 12 regions of interest across the tumor, the normal, the stroma. And this is just a snapshot of at the bottom, the graph just shows the variation in individual markers per region of interest. So we can see whether a tumor-rich region is high in cytokeratin, or just to understand whether the assay worked or not initially before we go into the multi-marker work. And the panel of antibodies we looked at are on the right-hand side here. So there's a number of controls, a number of housekeepers, and the rest of the markers span immune activation, immune cell profiling, IO drug targets. So a number of novel markers as well in the STING, VISTA, IDO1, which are really interesting markers, I think, in the IO space. And some pan-tumor markers as well. And we were able to do this across multiple tissue sections and generate heat maps essentially for each tissue section. So where we're able to measure a tumor signature, an immune signature, and an infiltrating edge or a mixed signature where there is overlap between tumor and immune regions together, and just to get an idea of what cells are there and then the certain marker expression levels. And we're able to do this from about 20 microns all the way up to 400 microns. So it depends on what your region of interest is and what you're looking at, whether it's in the global level or you want to go down into the sort of the 10 cells or profile 20 cells at a time. And this is just a snapshot of what we're able to do. So this is the 40 markers measured across this one tumor, just showing different expression levels, just to see what sort of data we're able to get. And on the right, what we see is the variation in three markers which are being looked at in the IO space of VISTA, STING, and IDO1. And you can see the variation across three head and neck tissues for these three markers. So they're involved in immune regulation, and we're trying to look at this in greater depth. So not just the PD-1, PD-L1, but what about the other markers? And then spatially, where is VISTA expressed? Where is STING expressed? Where is IDO1 expressed? And how does that associate with disease progression? And this is another snapshot of the variation across those 8 tumors for the tumor, immune, and mixed ROIs, and the variation across those markers going across the chart there. And in this slide, we're able to compare tumor regions versus immune regions and see what sort of cells are expressed there spatially, what cells are within the tumor. It's not just about the CD8 cells within the tumor, but I think what other cells are there. I think some of David Rim's work showed that CD68 cells in the tumor were predictive in melanoma. So I think, not just looking at the CD8 cells, but what other cells are brought in, the NK cells, what are the FOXP3 cells could be really interesting. So we're able to compare different regions of interest and see what sort of markers are expressed, what sort of immune cells are recruited into the tumor or excluded from the tumor. And in the bottom, we're able to measure the infiltrating edge versus an immune-rich area as well. So the same sort of analysis, just giving us a snapshot of the signal within those areas. So validation is always important, and this was quite challenging for us because we were looking at 50 or 60 immuno-oncology markers spatially and how we could then validate this data. So we looked at using a smaller multiplex IHC panel. So in this case, 4 markers, but we built it up to 6 markers at the moment, using the Carcinoma Opal staining kits, so CK, CD8, PD-L1, and DAPI were done on similar regions of interest. So in the top, you can see an H&E section demarcating the tumor, and then that was profiled on the nanostring and the Opal systems and assessed for similar regions of interest to see how the expression levels varied. And we were able to find that both systems were quite concordant with what they were the staining across both systems. And this was reaffirming to us, and we're doing this in greater depth at the moment. So this is just some analysis between the total set of samples done. So the patient clinical pathological findings are on the left-hand side. So 2 patients received Nivo and 5 received Pembro in the metastatic setting. And we were able to compare the marker data to survival outcomes. So whether they were progressors, whether they were non-progressors, so NP demarcates non-progressors and PD demarcates progressive disease. And what was really interesting from this initial pilot data set was that CD8 cells weren't predictive of outcome. And this sort of goes against the current literature, but I think this was interesting because spatially we could assess this. And what's interesting was on this next slide, we could see that there were a number of markers that were predictive of progressive disease. So CD44, STING, CD66, phospho-AKT, and PTEN. So now we're looking at these markers at a greater depth and really trying to understand spatially where they are and what other cells are recruited within these regions. And PD1 and PDL1 at the top of this graph here, and granzyme B too. So this is just some preliminary lung cancer data where we did a tumor microarray, so 92 cases with tumor and matched normal, and looked at it on a 60-plex IO panel, again, protein. And what we wanted to do was use the masking function. So this is where you're able to take the pathologist's demarcation of the tumor and normal regions, and then you're able to draw within it. So in this, in the bottom right-hand corner, you can see that you can make a tumor mask, and you can liberate the signal of that. And when you do the inverse, you get the tumor microenvironment signal, which is, you get a total signal per core for the tumor and the microenvironment data. And this is what we're trying to attempt with the lung TMA. And this data is really, really new. So I'm really excited about this data. And just a quick snapshot of what we're able to do when we're able to compare the stroma versus the healthy cell. Each core had a matched normal tissue as well. And we're able to see the number of markers that are highly expressed, which are significantly found to be present between the healthy and tumor versus the stroma and tumor as well. And VISTA comes up again when you compare the tumor versus the stroma, CD44, CD45. So there are a number of markers here that are highly significant that we're trying to look at in a greater depth. And at the bottom, that's just a heat map comparing the tumor, the stroma, and the matched paired normal. So this is really preliminary data, but it's really exciting that we're able to do this from a tumor microarray. And we're hoping to really use that geomasking function to get 2 signals or 2 signatures per core, and then we'd be able to measure it across a number of TMAs. And I think this is really powerful. In this case, this was in non-small cell lung cancer. So in summary, multispectral spatial proteomic profiling characterizes the tumor microenvironment in HNSCC patients treated with IO. CD8 T cells were not found to be predictive outcome to immune checkpoint therapy. There were a number of other markers, CD4, CD68, CD45, 44, and 66B, were found to associate with progressive disease in a preliminary cohort of patients. And in one patient, there was a low to intermediate TMB for that tissue section. And so the DSP data revealed an increased PD1 and low PDL1 in the tumor ROIs. So comprehensive analysis of the TMB and spatial TME, so tumor microenvironment, may in combination predict outcome in the future. And these applications, I think, can be extended into CTC analysis and organoid cultures if you can get those cells on slides, and then you'd be able to profile them. So I think that's a really novel angle that I'm looking at as well. With that, I'd like to thank you for listening to the talk and to acknowledge the funding agencies that contributed to this study, as well as the patients that gave their valuable samples. And I'd like to thank the team in Brisbane that assisted with this work as well. Thank you.
Arutha, thank you. That was really fantastic. Very exciting talk. We have a number of questions for you now. So the first one is, how were regions of interest chosen in this study, and is whole slide analysis available?
So regions of interest were chosen based on a pathologist review of an H&E section, and then we would stain that section and visualize it based on cytokeratin, CD8, DAPI, and one more color, CD3, CD4. And then we would match that up against an H&E section and then choose different regions of interest across tumor, normal, stroma, and measure that.
So another question here relates to kind of how you use tissues in this analysis, and it's how do you compare the use of full-face tissue sections versus tissue microarrays for DSP analysis? Which did you prefer? And perhaps you could comment on your results from those two approaches.
I think that's a great question. So we did full-face tissue sections for the head and neck study, and now we're going into tissue microarrays and doing that in head and neck and lung cancer. So I think with TMAs, you're able to profile a lot more samples, but the full face tissue sections obviously have their value in that you're able to get a full appreciation for the tissue section and not just a core biopsy.
Arutha, I have another question for you here, and it's an important one, which is, how do you validate DSP data?
So the DSP data was validated using the Opal Perkinoma solution. So the multiplex IHC, you could do this using imaging mass spec or just multiplex IHC. And we did it at 4-plex, but I think there's the scope to increase it to 6-plex assay. And ideally, you do the same regions of interest or as close as possible to see how that looked.
So, Arutha, another question here, which is really kind of asking you to discuss the challenges involved in the spatial analysis that you've described to us today.
Yeah, that's another great question. So I think the challenges in spatial analysis, I think you need to be really clear about the question that's being asked, whether it's a clinical question, whether it's a research-based question, because the data that you get back is extensive and you could be under that for months. So really knowing what you want going in will probably dictate what you get coming out. And that's really important going in. And I think there's a lot more data that comes out of these sorts of studies, and they're great for sort of hypothesis generating and questions like that. But really being specific about the question that you're asking is really important, because the volumes of data you get back did keep you under for months on end.
And finally, Arutha, your last question is, how did you choose the visualization markers that you used for your study?
So the visualization markers were chosen based on some simple stains such as cytokeratin, CD8, DAPI, CD3 or CD4 cells. We really just wanted to demarcate the tumor microenvironment from that of the tumor and compare that to the H&E section. And those are sort of universal markers that are used. So I think the takeaway message from this initial analysis and this data set is that digital spatial profiling is a really powerful technology, but I think we need to be really careful in designing our experiment and being really careful in choosing what our research question is, because the data sets that we get out are really, really massive. And really being able to sit around and have a pathologist on board, a clinician on board, a number of scientists on board, and being able to interrogate that tumor section or that tissue microarray is key. And it's a really exciting space, I think. and it will likely lead to the clinic. A lot of this work is being done with human specimens. So I think it's a really powerful technology to really understand the tumor microenvironment. There's a lot of information that we can get out of it. So initially it was just, are CD8 cells excluded from the tumor regions? Are they infiltrating the tumor? But the level of depth of information that we cannot get at the protein level, and that's not even going into the larger panels that are being done at the RNA level, such as the cancer transcriptome atlas work, which we're doing at the moment. And that's really powerful, I think, going forward, getting the complement of the RNA data plus the protein data, associating that with outcomes to checkpoint therapies, be it in melanoma, head and neck cancer, non-small cell lung cancer. It would be a powerful sort of a companion diagnostic tool to assist in selecting patients that are likely to benefit from immune checkpoint therapy, and in that way target therapies to those patients that are likely to receive the greatest benefit. And with that, I'd like to thank everyone for listening to my talk.